Let this be the last urinalysis objective anyone ever has to do. It seems like we've had a urinalysis every couple of weeks since block 1, and every time we get a new one, someone has a question like, what does that color mean? Or is the cloudiness indicative of the exact type of infection we have? Or is there a way we can narrow down our differential based on this urinalysis? I hope I can answer all of these questions right here and no one ever has to ask again. A urinalysis is basically broken down into 3 parts. You got the physical exam, the chemical exam, and the microscopic exam. Okay, so it's just like it sounds. The physical exam, you're just going to look at stuff. What color is it? Is it turbid? Is it cloudy? What's the specific gravity and what's the pH? Now, some texts will actually add to this smelling. You want to smell it? I don't know if a lot can be gained from that. What you're going to see, though, here in a minute is that some of these chemical tests can actually have some false positives. And so smelling this can actually, if it's putrid, you can kind of increase sensitivity and specificity. Then in the chemical test, you're going to look for blood, or what's really going to be either hemoglobin or myoglobin. You can't tell for sure if it's blood until you look under the microscope, but you can detect hemoglobin and you can also detect myoglobin on the same thing. So it could be an indication of lysed blood cells or lysed muscle tissue, myocytes. You're going to just check for proteins in general. Check for glucose because if there's diabetes mellitus or any type of filtering problems, then you're going to have glucose in the urine. Check for ketones. It's going to indicate a lot of metabolic things going on in the body. Urobilinogen, bilirubin, white blood cells, and nitrates going to look for infection. Then on the microscopic exam, cells, cast crystals, and then bacteria, yeast, parasites, just any type of invader cells. So what do all of these things mean? All right, the first thing you're going to look at on a physical exam is the color. The normal color can be anything from straw-colored all the way up to amber. I really don't know if that's straw-colored and that's amber because I'm color deficient. I can only see about 1/3 of the spectrum. In fact, any color that can also double as a girl's name is not really a color to me. Likewise, the only fruit that is a color is an orange. A kiwi, for example, is not a color. But what these represent, so a straw-colored urine would indicate that a person is well hydrated, or at least that their urine is dilute. This would indicate that their urine is concentrated, usually because of dehydration, but not always. What gives your urine its color? Well, there are these highly conjugated molecules called urochrome. And so I guess technically you could do a spectral assay of different concentrations of urochrome and figure out what normal urine really could look like under a photospectrometer. I'm not that into it. The concentration of urochrome varies with the specific gravity, and so the higher the specific gravity, the more amber, the lower the specific gravity, the more straw-colored. Abnormal colors of urine include everything from colorless, which I don't know why they call it a color. It's not a color, it's the very opposite of a color, but it's colorless, meaning overhydration typically. You can have red urine, orange urine, brown urine, even black urine. If I saw black urine, I'd freak out. A green-blue urine and a yellow urine. What do all of these colors mean? A lot of these colors are normal, but they also indicate other things. So, for example, you can have yellow urine, and if that yellow urine could be normal, or it could mean that you have excess riboflavin. And so the big category that all of these colors fit into is you can put them into proteins can change color, drugs, a lot of prescription drugs will change the color of the urine, metabolic diseases, blood or heme in the urine, as well as myoglobin, and then the food that you eat, beets and rhubarb, for example, can change your urine to red. Giving you a huge list of all of these things is pointless. In fact, it's in my study guide if you ever need to reference it, but I actually would point you to Campbell Walsh Urology 10th edition. It has an almost exhaustive list of colors and what they mean. But because yellow can mean like 3 or 4 different things, none of these things are going to be specific or sensitive. They're just going to lead you in the right direction. You're going to give you more direction and where to look. For example, brown. Brown is really indicative of porphyria. But you also get brown pea whenever you eat fava beans. And if you're taking nitrofurantoin for a UTI, you can also get brown pee. So really, it's not going to help you a whole lot. It's just going to point you in the direction. So what does turbidity mean? Most of the time, it is not an infection, but it is phosphaturia. If your urine has a high pH, then it's going to precipitate phosphate, and you're going to urinate it out, and it's going to be cloudy. The second most common cause is due to infection, and you're going to get a turbid color because of the white blood cells. Now, how do you tell these two apart? There's two ways, actually. You can do the vinegar test, which is basically put acetic acid on it, and if it goes away, then it was phosphate. The phosphate was precipitated in the alkaline urine, and then the acetic acid redissolved it. The other way is to smell it. If it smells pungent, it's an infection. There are 4 other very rare causes of a turbid urine. You can have lipiduria, which is high lipids, it's just like it sounds, hyperoxaluria, and hyperuricosuriria. The 4th one, I'm just going to have to draw it for you. So here you have your kidneys, ureters, bladder, urethra, and then you have here this uncommon little lymph node that for some unknown reason it is connected to the urinary tract. So whenever you have a lymph node connected to the urinary tract, you can get lymphocytes in that urine even without an infection, and it will cause it to turn turbid. Okay, now specific gravity. Before I talk about what specific gravity is telling us, I have to basically point out something. Osmolality is a better measure of what we're looking at than specific gravity. However, you can't measure osmolality on a dipstick. You have to have an osmometer. So for using a urinalysis dipstick, specific gravity is usually all that's measured. Now the ranges for specific gravity are going to vary by source. Most of our cases, we say that the normal range is between 1.005 and 1.030. The textbooks say that anything between 1.001 and 1.008 is considered dilute. And they say that anything between 1.020 and 1.035 is concentrated. Now there's also this magical number, I don't know where it comes from, but 1.010, if it becomes fixed at this number, according to the textbooks, it's a sign of renal insufficiency. I don't know how long fixed means, the textbooks don't say that. So if I measured it today at 1.010 and then I measured it again tomorrow and then the next day, would 3 days be long enough? I don't know. Nobody knows. The most common causes to have a decreased specific gravity is increasing fluid intake, number 1, diuretics, number 2, and decrease in renal concentrating ability. The most common reasons for an increased specific gravity is having a decreased fluid intake, dehydration, which I'm referring to sweat, vomit, and diarrhea, diabetes mellitus, so you're urinating out sugar, glucoseuria, so that would make sense that the specific gravity would be higher with sugar inside of it. Symptom of inappropriate antidiuretic hormones, so basically your body is forcing you to concentrate the urine. Having treatment with IV contrast and also treatment with dextran can increase the specific gravity. Then the last 2 things I want to mention are the heart and the kidney themselves. So over here, if we have a decreased blood flow to the kidney for any reason, say for example renal artery stenosis or heart failure, for example, then you can have an increased specific gravity. On the other hand, decreases in specific gravity are associated with damages to the kidney, so the common nephritises, pyelonephritis, interstitial nephritis, acute tubular necrosis. And this all feeds back into number 3, so the renal concentrating ability is decreased whenever the kidney is damaged. So if you want a quick and easy way to remember this, with the exception of fluid intake, this is everything that happens to the kidney, while this is everything that happens before the kidney. And then the last thing I want to point out is that the 2 different diabetes are on different sides of this thing. So diabetes insipidus, this has to do with command signals going to the kidneys saying, hey, I want you to do this, whereas this has basically to do with the blood sugar, so this is before the kidneys get involved. I would just like to point out that this week we had a very low specific gravity. It wasn't dangerously low, it was 1.008, but this decreased specific gravity fit very well with our picture of damage to the kidney. Okay, let's talk about the pH of the urine. The physiologic range for pH is anywhere from 4.5 to 8.5 in the urine. Now, anything from 5.5 to 6.5 is considered neutral. From 4.5 to 5.5 is considered acidic. And from 6.5 to 8.5 is considered alkaline. In our case, we had a pH of 8.5. Why was that? Well, before we go into that, first I want to discuss exactly what pH means. Typically, the pH will follow the body. It'll follow the serum pH. That's because if the pH goes up in the serum, then the kidneys are like, wait, it's too alkaline in my body. I'm going to start secreting some of this base. And so it brings its pH up to start getting rid of the extra base and bringing the pH in the body back down. And vice versa. Now, what are the exceptions to this rule? Renal tubular acidosis type 1 and 2. This is where the kidneys unnaturally secrete bicarbonate. They secrete it whenever they shouldn't. So your serum pH will be unnaturally low while your urine pH will be unnaturally high. This is not the problem that we had today. Some bacteria secrete urease. So here we have our molecule of urea. When it is near 3 molecules of water in the presence of urease, it can be converted to carbon dioxide and 2 molecules of ammonium hydroxide. And if you remember from organic chemistry, whenever we have ammonium hydroxide, there's going to be a nucleophilic attack where these pull off the hydrogen. The extra electrons are going to stay on NH3, and you form water plus NH3. And NH3 is a base. It can pull off more protons and be stable in the ammonium form. I bet you're wondering which bacteria can do this. Because if we can see the pH of the urine, if the urine pH is high and the serum pH is normal, so I'll put blood, it's normal, then this would indicate a possible bacterial infection with a urease positive species. This is especially so when you combine this with other tests such as the nitrite test and the leukocyte esterase test. First aid gives this mnemonic: Chuck Norris hates punks. So anything in blue is what really matters. The rest of it, just forget it. So the species that first aid lists are Cryptococcus, which is a fungal infection, Helicobacter pylori, Proteus species, Ureoplasma, which is a cousin of Mycoplasma, Nocardia, Klebsiella, Staph epidermidis, Staph saprophyticus. However, a quick search of Google and we yield 2 other species that aren't listed in first aid, which are Campylobacter and Brucella. In fact, Wikipedia also mentions Morganella, Providentia, and possibly Serratia, all being enteric bacteria like Proteus and like Klebsiella. However, Wikipedia does not specify whether these are found in the feces or the urine or both, so I'm questionable whether they're relevant. Why is pH a big deal besides the fact that it can indicate renal tubular acidosis? There's more to it than that. Besides the fact that it can indicate infection, a high pH can precipitate calcium, magnesium, ammonia, and phosphate. We mentioned phosphate earlier. So if you don't want to get stones from these things, you don't want the pH to get too high. And what if the pH gets too low? In this case, you can get the precipitation of cystine and uric acid. This is especially important in stenosis and in other causes of gout, namely anything that causes a high purine degradation. Okay, that wraps up the physical examination of the urine. Let's take a look at the chemical examination. First, we're going to look at the leukocyte, esterase, and nitrite. We're going to take a look at these together because they both together point to bacterial or microbial infection. If they're redundant, why do we need both of them? Well, because the sensitivity and specificity of either one by itself is not extremely good. For example, nitrites have a decent specificity. So the specificity is around 90%, but the sensitivity is anywhere between 35 to 85% depending on the source you look at. So if you get a negative nitrite test, yeah, you're great. But if you get a positive one, you still got to look at it under the microscope to be sure. However, when you add in the leukocyte esterase test, sensitivity and specificity goes up tremendously. However, urologists still recommend to take a microscopic examination of the urine. And in most cases, if you're dealing with a drug-resistant bacterium, you're going to want to culture it and grow it so that you can test its sensitivities. We'll start by looking at leukocyte esterase. This specifically identifies the presence of a neutrophil. A neutrophil will secrete leukocyte esterase. This is great. Neutrophils there indicates an infection. So how do we know if the neutrophil's there? We test to see if leukocyte esterase is there. So on our dipstick, we're going to have some indoxyl carbonic acid. In the presence of leukocyte esterase, that will get converted into indoxyl. However, how do we know the indoxyl's there? So for the last step, we're also going to have a diazonium salt. The diazonium salt will start off reduced. In the presence of indoxyl, it will become oxidized. Now you remember from organic chemistry that diazo dyes, these are basically nitrogen-based dyes that were used extensively back at the beginning of the industrial era. Depending on their oxidation state will depend on what color they are. Well, for a portion of them anyhow. And so when we get color change, we know that there are neutrophils present. And as a rule of thumb, for the diazonium reaction, you want to wait 5 minutes. Some of the main reasons you're going to get a false negative on this, if your urine has too high of a specific gravity, glucosuria, the presence of urobilinogen, if your urine color is altered by medication, then it can make the test impossible to read, and then if you ingest high amounts of ascorbic acid. The overwhelming reason to receive a false positive is contamination of your sample. That's why you have to follow protocols for the urine collection specifically. Most protocols say for a guy it has to be a midstream catch. If they are uncircumcised, they need to retract the foreskin. Everything needs to be wiped down clean before you start urinating. Everything has to be done exactly. For women, even sometimes there has to be catheterization. And when there isn't catheterization, there's other weird protocols y'all have to do as well. So the nitrite test works like this. Basically, many gram-negative bacteria produce an enzyme called reductase. Now, notice I said many, not all. That's why you can have a negative nitrite test and still have a UTI. That's why the sensitivity is between 35 and 85% when you use nitrite test alone. So your blood, your urine actually has nitrate in it. Nitrate being NO3 negative. If this reductase molecule is present, then what will happen is the nitrate will be converted into nitrite. This doesn't happen naturally in your body, especially not in your kidneys or urinary tract, and so if you have nitrite in your urinary tract, it definitely indicates the presence of reductase, which indicates the presence of a gram-negative bacteria. How are we going to know, you ask? How are we going to know if the nitrite is present? Similar to the leukocyte esterase test, we're going to have a diazo-producing reagent. In the presence of nitrite, it's going to be reduced to a diazo dye. Bam! Color change. It's usually a red dye. If you have less than 10 to the 5th organisms per milliliter, this test usually isn't as accurate. And as with the leukocyte esterase, the major problem with false positives tends to be from contamination. And false negatives not only have to do with the number or abundance of the bacteria, but also whether or not that bacteria is a specific gram-negative that produces the reductase enzyme. And unfortunately, try as I may, try as I might, I wasn't able to find an exhaustive list of all of the gram-negative bacteria that have reductase, nor was I able to find all of the bacteria that don't have reductase, so you're just going to have to live with that. I know it's heartbreaking. Okay, the next test is the blood. A positive blood on a dipstick can indicate one of three things typically. You can have actual blood in the urine, you can have hemoglobin in the urine, or you can have hemoglobin's cousin, myoglobin, in the urine. This test basically takes advantage of the fact that iron has both a peroxidase cycle and an oxidase cycle. But for this test specifically, we're using the peroxidase cycle. Okay, let me back up just a little bit. What is hemoglobin? Hemoglobin is heme plus a globin. Seems simple, right? So what is heme? Heme is basically iron plus protoporphyrin. So we're taking advantage of the fact that both hemoglobin and myoglobin contain iron in the center of their structure. So here we have the peroxidase cycle. It starts off with iron 3, and then you'll have hydrogen peroxide will be converted into water, and one of the oxygen molecules and an electron will be added to the iron to make it iron 4. Then you can take an electron and a hydrogen ion and add that to the same molecule, having this complex hydrogen, then add another electron and another proton, and you'll get water, and you'll return to iron 3. But where do these electrons come from? So in order to figure that out, we got to talk about how this test becomes positive. So we have all these electrons, and we don't know where they came from. Okay, so let's take a look. We have something called a chromogen. And what we're doing is we're oxidizing this chromogen and basically stripping away electrons from it. And by doing that, we get a color change. Okay, now let's talk about the test strip itself really quick. So we have this test strip, and when we put hemoglobin on it, we get a large area that's positive. Now this is true for hemoglobin and myoglobin. However, if we put erythrocytes on it specifically, they're going to stick to the test strip and lyse on the test strip, making small dots. And the small dots will appear there until the number of erythrocytes increases beyond 250, and at that point they'll start to coalesce. So if the erythrocyte number is low, you can actually distinguish between whether you're getting hemoglobin or myoglobin and actually hematuria. Now another question we have to ask ourselves is the blood that we're seeing nephrogenic or urogenic? Now typically blood can get in through the kidney by having damage to the glomerulus. And when this happens, you not only get increased blood, but you get increased protein because protein is no longer being filtered out. However, if the source of the blood is urologic, then you're usually going to have a normal amount of protein. In addition to protein, you also typically get casts, which can be seen under microscopic examination. Also, if the erythrocyte came from glomerular damage, typically whenever you look at them under the microscope, they're going to be dysmorphic. However, anything further downstream than the glomerulus, which means it can be interstitial, it can be urologic, anything downstream from the glomerulus, they tend to be a normal shape and size. Now these three things are true, so we're looking at glomerulonephritis, and we're saying that typically we should see these three things in addition to blood. However, this is only true 80% of the time. 20% of the time, the only thing you see is blood. You don't see any of these other findings. Now the workup for hematuria is pretty extensive, and for that, I'm going to refer you to Chapter 3 of Campbell-Walsh Urology 10th edition. Now let's move on to protein. Protein is detected in the urine using a dye called tetrabromophenol blue. The biggest deficit with this is that it particularly detects albumin over other proteins. Protein in the urine can be broken down into three major classes. There can be glomerular damage, there can be tubular problems, and there can be overflow. The hallmark of glomerular damage is a high albumin in the urine. With tubular problems, you get a lot of low molecular weight proteins in the urine. This is due to a failure to reabsorb those proteins, and it can be caused by high glucose in the urine, amino acid urea, phosphaturia, and then Fanconi syndrome. The idea behind this is that several small proteins get filtered through the glomerulus. However, they get reabsorbed in the tubules of the kidney. Whenever the tubular function of the kidney isn't working properly, then you get these low molecular weight proteins in the urine. Overflow is analogous to diabetes, but with proteins instead of sugar. The most common cause of this is multiple myeloma. Large numbers of immunoglobulin light chains are produced and filtered through the glomerulus, and they overflow. They saturate the number of receptors that are there to reabsorb them, and so you get them in the urine. Usually special tests have to be done to detect these lower molecular weight proteins, and then you also usually have to do electrophoresis with immunologic staining in order to tell whether it was albumin or a light chain immunoglobulin, or if it was another low weight molecular protein. Testing for glucose and ketones. Typically, all of the glucose that gets filtered in your urine or through the glomerulus into the kidney gets reabsorbed by the kidney. Once you hit 180 milligrams per deciliter, once you get that concentration being filtered, you have saturated all of the receptors in the kidney to reabsorb the glucose. And so at that point, you start getting glucose in the urine. This happens because the blood glucose is just really dang high, such as with diabetes mellitus. Ketones are typically present with diabetic ketoacidosis, and so basically what happens is when your body thinks there isn't enough glucose. And why would it think that? Well, it thinks that because it isn't listening to insulin. It's insulin insensitive, or else there is no insulin. So type 1 diabetes, there is no insulin to tell the body, or type 2 diabetes, the body can't hear the insulin. So your body thinks, oh, there's no sugar, because it can't hear insulin screaming, hey, your sugar's out here waiting for you. And when it thinks there isn't enough sugar, it starts breaking down fats. One of the byproducts of fats is called ketones. The three ketones are acetoacetic acid, acetone, and beta-hydrobutyric acid. So in addition to diabetic ketoacidosis, you can also get ketones in the urine from starvation. You can get them in the urine at times from pregnancy and at times due to rapid weight loss. Biliruben is a byproduct of the breakdown of red blood cells. First, in the spleen, you get the breakdown of the red blood cells, and you get this unconjugated bilirubin. It's high molecular weight, and it's bound to albumin in the serum because it's not water soluble. It then moves into the liver, and the hepatocyte will conjugate it to glucuronic acid. So conjugated means that it's tied to glucuronic acid. This becomes low molecular weight and becomes water soluble. This conjugated bilirubin then gets secreted into the GI system via the bile duct. In chronic liver disease, or when there's an obstruction of the bile duct, then you can get conjugated bilirubin in the urine. It'll back up, get in the bloodstream, and go into the urine. On the other hand, unconjugated bilirubin, because it is not water soluble and because it's bound to albumin, it is not seen in the urine on any conditions. Now, when this conjugated bilirubin gets into the GI tract, the bacterial flora will turn it into urobilinogen. So now you have urobilinogen in the intestines. Then, 50% of this gets excreted. So 50% is excreted, and then the other 50% is reuptaken through the hepatic portal vein into the liver. Occasionally, a small amount of this will escape the liver reuptake and be found in the circulation, and you can find a small amount in the urine. However, when there's an increased production of bilirubin and therefore an increased production of urobilinogen, you can get increased amounts in the urine. Why would you have increased production of bilirubin, you ask? Well, you'd have an increased production if there's a hemolysis going on. Then also if hepatic disease is taking place, then you have a failure to reuptake it efficiently when it's being sent back from the GI tract. So that would cause an increase in urobilinogen seen in the urine. Decreases in urobilinogen can be seen whenever there's antibiotics that mess up the microbial flora of the GI tract, and also obstructions of the bile duct that prevent the urobilinogen prevent the bilirubin from getting into the intestinal flora and being converted.